Hi, my name is Nancy and my partner's name is Nazia, and we are going to do the experiment on fermentation of fruits by Saccharomyces cerevisiae and determining whether yeast or substrate is limiting reagent. Based on our research, we assume that sugar is limiting reagent, and if we keep on increasing the yeast amount in the experiment and keeping the sugar content steady at the same time, there will be one condition that there won't be any more sugar for yeast to ferment. So for the experiment, we'll be using this yeast and these glass bottles. We have sterilized these glass bottles using boiling water, and the apples we cleaned it with the hot water, and we will be using the mush of this apple. And these are the balloons that we'll be using to measure carbon dioxide. We will put these balloons on the top of each bottles, and then we will measure the carbon dioxide, and these centrifuge tubes we will be using for measuring the. For independent variable, we will be using three different variables: light, temperature, and time. For light, we will be keeping all our experiment setup in darkness, and for the temperature, it will be around room temperature, 28 to 30 degrees centigrade, and the time we will be keeping it at least for a week. Moreover, for the independent variable, we will be manipulating the yeast and sugar amount. So for dependent variable, we will be evaluating the production of carbon dioxide and the smell and the appearance.
Initially, all the apples were soaked in warm water for half an hour and washed properly, and then I am destemming them so that they don't interfere in the fermentation process. Then the crushed apples are poured in these bottles, 250 ml each, and the bottles are labeled like this: control, apple pulp, no yeast, with date and time. Then the yeast is put in different tubes for activation, and the yeast is set aside for 10 minutes for activation. 10 minutes has passed, so I am just mixing the activated yeast with the fruit pulp and then mix it properly by swirling. After that, I attached balloons on all the bottles, and the darkness is provided by a blanket. So let's start measuring carbon dioxide now. With regards to sensory results, the color of the pulp has changed significantly. There is liquid on the bottom, and the fruit pulp, the residue, it came on the above. The fragrance of this is quite fruity and strong. There is still bubble formation, so the fermentation process is still ongoing. For control, there was 30 ml formation of carbon dioxide, whereas for 0.15 gram of yeast, it was 35 ml, and for 0.30 gram, it was 60 ml, for 0.50 gram, it was 85 ml, and for 0.75 gram, it was 115 ml. After that, we boiled the apples for some time, like half an hour, because there was contamination last time. So after grinding, we have mush for our week 2.
So for the week 2 of our first trial, we have used a 50 ml mush of apple, 100 ml mush, 150 ml mush, 200 ml mush, and for the control 250 ml mush. And then we are going to pour activated yeast, which contains 2 gram of yeast with the using the water, and we are going to pour each of these tubes in each of these bottles, but not in the control.
After pouring the yeast, we shake the bottles well.
So after shaking the bottles, we in all 5 bottles we have attached the balloons to measure carbon dioxide on the top of each bottles like this. So these all 5 bottles we just kept it in dark and covered like this. Trial 1, week 3, we have used 0.6 gram of yeast in each three tubes, and then 10 ml of water in each, and then 3 grams of glucose, 3 gram of lactose, and 3 gram of sucrose. So for this one we have used two controls, and in each two control it's 10 ml of water, and for control number 1 it's 0.6 gram of yeast with 10 ml of water, and in another control 10 ml of water with 3 gram of sugar. In 3 gram of sugar it's 1 gram of each 3 sugars that we have used for the test.
After mixing all the contents, we put these balloons over the tubes and put the tubes in these containers, and we will wait for 5 to 6 days to measure the carbon dioxide content. After that, we use the same curtain to put the balloons with the container in the dark. For control 1, which was having only sugar, there is no inflation. For control 2, which have only yeast in that, it also did not show any inflation. For sugars, least fermentation was shown by lactose and yeast, followed by sucrose, and glucose mixture with yeast, it showed most of fermentation and carbon dioxide formation. So let's start with measuring carbon dioxide for lactose solution. It turned out to be 6 ml only. Now we are doing for sucrose and yeast solution. It came out to be almost 33 ml. And now let's do it for glucose solution. And the one with glucose, the balloon with glucose showed almost 85 ml of carbon dioxide production. The results are kind of expected, since the bottles with less amount of sugar, 50 ml, 100 ml, formed negligible amount of carbon dioxide. However, with 150 it formed significant amount, but it was very less as compared to 200 ml. When the sugar was present like in larger amounts for yeast, for fermentation, there was more carbon dioxide formation. For the control, there was very less carbon dioxide formation as the previous week. For 50 ml, it was like negligible amount, 0 ml of carbon dioxide formation. So we started from 100 ml of sugar container. It turned out to be 48 mLs. For 150 ml sugar containers, the carbon dioxide is 105 ml. Let's start for the container with 200 ml of sugar. For the container with 200 ml of sugar, it comes to be 160 ml of carbon dioxide in the balloon. For control, it comes up to be 10 ml of carbon dioxide formation. The control was contaminated since there was formation of these white colored colonies in them. However, no such colony was observed in any of other bottles. The color of control, which is this one, is also very different from the other bottles.